Smith, and I am presenting hemoglobin, or really the heme most definitely. Hemoglobin has 4 areas. There's 2 alphas and then 2 betas, and they're connected with using histidine and iron. There are 4 irons which will be connected and have the ability to connect to 2 oxygens each. There are 2 states. The R state, which is the rested state, is oxygenated, and then the T state, which we can think of as the 10th state, which is deoxygenated, where the iron in the heme group sits slightly outside the plane of the porphyrin ring, and it's puckered. You can see to the left, it's just, there's a bigger hole. It's just because the iron is too large to fit in the center. There's the atomic snap of oxygen binding to that heme, which you can see in the picture. The binding where oxygen binds to the 6th coordination site of the iron, and it changes the electronic state of the iron, making it effectively smaller. So remember, it becomes, the hole is not as big. As far as structural shift, the iron snaps to that place of the heme ring because the iron is attached to a proximal histidine residue. It makes the movement pulls the entire alpha helix of the hemoglobin subunit with it. And there is a cooperativity to this. The small shift in one subunit breaks iron pairs like salt bridges at the interfaces of the other subunits, making it significantly easier for the next oxygen to bind. And that's that transition from tense state to relaxed state. So T to R. When looking at it, binding capacity, lungs versus capillaries, hemoglobin has a genius shape, which is that sigmoidal S-shaped curve, which allows it to be kind of a greedy soak in the lungs and a generous giver in the tissue. So look at the very far left, it's in the lungs, to the right in the capillaries. In the lungs, the partial pressure of the oxygen is just very, very high. Hemoglobin nearly becomes 100% saturated because of the cooperativity mentioned a moment ago. Once the first few oxygen molecules hitch a ride, the R state dominates, and the heme group has a very high affinity for oxygen. In the capillaries, that changes. The partial pressure completely drops. Not completely, it just drops. Hemoglobin enters the steep part of its curve. Here a small drop in the pressure of oxygen causes a massive release of oxygen into the surrounding tissue. The affinity decreases, and the protein shifts back from the 10th state to dump oxygen, back toward that T state to dump oxygen where the cells need it most. So your surrounding tissues and everything. The carbon dioxide connection, pH and the Boer effect. The metabolic exhaust of your cells is carbon dioxide, and it plays a direct role in how much oxygen your hemoglobin releases. The carbonic anhydrase reaction is just when carbon dioxide enters the blood in the capillaries. It reacts with water, and that's catalyzed by an enzyme. And you can look at the image to your left, and it kind of shows what's going on. RBC stands for red blood cell. So in the plasma tissue, and in the acidification of this, the reaction just produces much more protons, those hydrogen atoms, which lowers the pH of the blood in the capillaries, making it thus more acidic. The Boer effect is just when these protons bind to specific amino acid residues on hemoglobin, and that stabilizes the T state or 10 state, further lowering hemoglobin's affinity for oxygen. And this, just the presence of carbon dioxide, effectively pushes those oxygen off the heme into the tissues, which produce that carbon dioxide and helps them receive the most oxygen. Then you just have happy blood cells from there. Like I said, hemoglobin can be described as almost like a greedy sponge, but due to high pressure and high pH but once it gets to the capillaries, it's more like a generous donor. Like, here you guys go. Here's all this oxygen I have. And that's triggered by the acidic environment in the tissues created by carbon dioxide. But that's all. Thank you for listening.